Okay, we're now going to move on to the negative-stranded RNA viruses. And before we talk about any of these viral families specifically, I want to let you know, again, that a negative-stranded RNA virus is a copy of the message, which means that it has to carry with it in the virion an RNA-dependent RNA polymerase, okay? Has to carry it with it. Our cells do not have that. We make RNA from DNA. Now, the positive-stranded virus gets around that by making it as soon as it gets into the cell. The negative-stranded virus has to bring it with it in the virion, okay? Now, there are quite a few viral families here, but you're going to see that this section moves kind of quickly because a lot of them, I'm going to tell you, are not really high yield for the boards, okay? Because we don't see them very commonly in the U.S. Now, the paramyxoviruses are a group of high yield viruses. This covers mumps, measles, RSV, parainfluenza, so again, a major viral family. In the rhabdovirus family, the only virus we really have to worry about there is rabies virus. The filoviruses, again, here's my little Ebola virus guy, the lasso-shaped virus. The filoviruses, unfortunately, because again, they're a little bit cute, are low yield for the boards. So we're not going to talk about them in any great detail. We are going to talk significantly about the orthomyxoviruses because this is the flu virus, and there's a few things that we really need to point out specifically about influenza viruses. The bunyaviruses, the only one we're really going to focus on is hantavirus. Arenaviruses, Lassa fever and LCMV, extremely unlikely. We really don't see very many infections with those. So we're going to kind of just mention a few things about the arenaviruses. Now, the negative-stranded RNA virus life cycle, we're using rabies virus as an example, and you should know that because rabies virus is the only virus that's bullet-shaped. You can see that here, okay? So the virus gains entry into the cell. It attaches, like we've talked about, has to get in, penetrate the cell, uncoat. Okay, so that's what you're seeing here. We have attachment and penetration. We have uncoating of the virus. Okay, and now what has to happen, the virus is carrying with it, you see this little dot here? That's representative of the RNA-dependent RNA polymerase that all viruses have to take with it in the cell. So now what's going to happen is that negative strand of RNA with the RNA-dependent RNA polymerase is going to get transcription into a positive strand, okay? That positive strand does two things. Number one, it functions as a message, so we're going to start to get protein synthesis. The other thing it does is it functions as a template to make more genomic RNA, all right? The virus then will eventually, once we get structural and non-structural proteins, will start to assemble and eventually push through and bud from the host cell, okay? So the major difference here between the negative-stranded RNA viruses and the positive-stranded RNA viruses is that the negative-stranded virus carries with it the RNA-dependent RNA polymerase. The positive-stranded RNA virus makes it as soon as it gets into the cell, so it doesn't have to carry it, okay? So that's the general synopsis for the life cycle of the RNA viruses. All right, some of the abbreviations that you're going to see within this section are H, which often stands for hemagglutinin, N for neuraminidase, and occasionally with the influenza terminology, which is where you're going to really see those, they'll sometimes call them HA and NA for hemagglutinin and neuraminidase. M typically stands for the matrix protein, F for the fusion protein, and P for that polymerase, the RNA-dependent RNA polymerase associated with the virion.